In this lesson, we'll continue our discussion of the cellular level of organization by taking a look at how substances move in and out of the cell. This is known as membrane transport. There are a number of different types. We're going to focus on diffusion in this lesson. So before we look at diffusion specifically, let's just review for a moment. We know that from previous lessons, the cell membrane or the plasma membrane, one of its functions is to be a barrier, right? Remember to separate the fluid on the inside of the cell, the ICF, from the fluid on the outside of the cell. However, think about this, right? Cells are living. They need to have nutrients. So nutrients have to cross that cell membrane. They have to come in so that the cell can survive. Things like carbohydrates, amino acids, ions. And then at the same time, as the cell is going through its different metabolism, it's going to produce different things, maybe proteins, it's going to produce waste products. Those have to leave the cell. Okay, so the cell membrane does need to allow for this type of movement. Now, we do use a term that kind of describes what moves in and out of a cell. That term is permeability. There are three different types of permeability. The first one is a membrane that is impermeable, meaning that it doesn't let anything come in or anything get out. The second one is freely permeable. That's like the opposite. That lets anything pass through, right? So anything's allowed to move through the cell membrane. And then finally, somewhere in between that, and this is most cell membranes, there is selectively permeable, where some things will move in easily while other things can be restricted. So let's take a look at a diagram to demonstrate this. We'll take a look at an impermeable membrane first. So there's three pictures here showing in blue the cell membrane. So here's a light blue one. This one is going to be a darker blue, and then this was kind of like somewhere in between. Let's take a look at the darker blue first, the impermeable. So this is a membrane that's showing you proteins are being blocked. That little double line right there, guys, means that something is being blocked. So the proteins are being blocked from moving in this membrane. Lipids, ions, water, carbs like glucose, right? They're being blocked from moving through the cell membrane. That you're going to call impermeable. Just the opposite. This membrane here, they kind of painted in light blue, very light blue. This is a freely permeable membrane, meaning that very easily proteins can move in, the lipids, ions, the carbs, the water, everything can move into the cytosol, which is in kind of yellow over here. I forgot to mention that there. This is the cytosol on the inside. So again, two opposites, freely permeable, anything can go through, impermeable, in this case, nothing is getting through. So somewhere in between the cell membranes, and this is most cell membranes, that they are selectively permeable, right? Certain things may be restricted, like in this picture here, the protein is being blocked. The lipids are being allowed to go through this time. Ions are being blocked. Water can move through. Carbs are being blocked, right? So the membrane has the ability to decide what needs to go or what can go through to the cytosol. Okay, so it's a good diagram kind of showing us the three types of permeability. Again, impermeable, nothing. Freely, everything. And then somewhere in between, I put it over here, right? Somewhere in between impermeable and freely permeable is selectively permeable. And most cell membranes are indeed selectively permeable. So what's really restricting, or what are some of the factors that restrict different substances from moving in or out? Number one could be size. If something that comes to the cell is very large, it may not fit through one of the membrane proteins that allow for transport, right? It may just be too big. If the substance coming to the cell, let's say, has an electrical charge that is repelled from the cell, like charges, right? Like charges repel. If the shape of the molecule coming to the cell doesn't fit into a membrane protein, that's going to be a problem. It's going to be restricted. And if a substance that's coming to the cell isn't lipid soluble, it may not be able to go through the phospholipid bilayer. So these are all factors that can restrict the motion, the movement. We'll take a look at how these are handled in subsequent lessons. So now, not all cells are the same on their permeability. Like I said, so some cell membranes are some some are freely permeable, and others are again are selectively permeable. What determines the permeability is basically the composition of the cell membrane. Again, we know from previous lessons that the cell membrane is made up of a phospholipid bilayer, and then embedded in that are the different types of protein. So based on the amount of phospholipids and the different types of proteins that are embedded, that will determine the permeability, right? So all of the cells are different based on their composition, right? Their percentage of phospholipids versus their percentage of proteins. One other thing before we look at diffusion specifically, energy. Sometimes movement across the cell membrane does not require energy. We say that is a passive type of transport, right? Passive process. No energy is required. Other times we need to expend energy like ATP to move something, say, against its concentration gradient. We'll talk about that in a little bit. That's an active process where we have to spend energy. So here's again an example of, or a diagram showing us the different types of processes as far as energy. We know that materials may cross the plasma membrane either through an active or a passive process. So we're going to see over the next couple lessons that the passive processes, as I said, don't require any ATP. That's going to include diffusion, which we're doing in this lesson, and then in future lessons we'll take a look at different types of carrier mediated transport. Some of them are passive, but you'll see, guys, some are also active, right? So we have a combination of passive and active types of carrier mediated transport, again in a subsequent lesson. And then there's something known as vesicular transport, also in a subsequent lesson, that's also active. All right, so the main thing right now for this slide is that a passive process does not require any energy to be expended, while an active process does. So now that we have some of those basics behind us, let's take a look at the first type of movement, which is known as diffusion, right? Here's the definition, the first dot right here. This is the net movement of a substance. We could think of it as a solute. Remember that solutes are the dissolved substance in a fluid, right? So this is the net movement of a substance from an area of higher concentration to an area of low concentration, right? So things are going to move from where there are more of these solutes, and they're going to move to where there is less of those solutes. In nature, we know that ions and molecules, they don't just sit stationary. Believe it or not, they're constantly in motion. There's kind of a natural movement, and as a result of this movement, the molecules in solution start to move around randomly, and that random motion causes mixing. If the molecules will bump into one another, and they'll kind of bounce off of one another, and over time it will cause a type of mixing. So this happens in a passive way. We don't have to put any energy in to get that motion. It's just a natural type of, they actually call it thermal noise. It's just kind of like a natural movement that our molecules have. Now, there's a term that I want you to know. It's called the concentration gradient. Very simply, this is the difference between the high and the low concentrations of a substance. Let's just say there's a cell membrane, and on one side there's a lot of, let's say, glucose. On the other side of the cell membrane, there isn't a lot of glucose. So the movement we're going to see will be from the area of high concentration to an area of low. That difference between high and low we call the concentration gradient. Okay, and as a general rule with diffusion, the concentration gradient will cause diffusion to be down the concentration gradient. We're always going to go from uphill to downhill. We're going to kind of go like down a mountain, right? Very, very easy. If you think about if you're on a bike and you're on a hill, you don't really have to pedal your bike to go from the top of the hill down. It just kind of happens without energy, right? You can kind of just coast down. That's what we're talking about. Um, Diffusion in the air and water tends to be slow as a general rule, and diffusion is most important over very short distances, which is what we're going to focus on. The cell membrane is not thick at all, so things are just going to diffuse from, say, one side of the cell membrane to the other. Okay, so on this slide, what I need you to know that are very important is the definition. Diffusion is the movement of substance or solutes from an area of high concentration to an area of low concentration. That is a passive process. Think of riding your bike down a hill, right? You don't have to pedal to go from the top of the hill to the bottom of the hill. You can just coast. And that's what's happening here. We're going to go from where there's more solute and we're going to roll down to where there is less solute. The difference between that area of high and low is known as the concentration gradient. So that basically describes, the steepness of the hill, right? A low hill would have a low concentration gradient, while a higher hill would have um, a steeper or a higher concentration gradient. So that's kind of what that's describing. And diffusion, the last kind of important thing here is most important over very short distances. So here's an example from our book where we see a beaker of water. And notice there's nothing in the water, it's pure water. So what they do here is they place a colored sugar cube, maybe with some type of food dye, and they drop it into the water. And that's going to establish a steep concentration gradient. So what do they mean by that? So in the sugar, guys, all here is where, I'm sorry, in the cube, this is where all the sugar is, right? So that's going to be the high concentration. What's in the water right now is no sugar, right? So that's going to create a concentration gradient from high to low. So notice once we drop the sugar cube in there, right, as the cube begins to dissolve, many sugar and dye molecules are in one location and none are elsewhere. So you can see that the sugar is starting to move away from where the cube was. See that little cloud right there? That's from that random movement of the molecules. And now we can see the food dye and the sugar starting to spread around. Over time, you can see that it starts to spread more and more, again, from an area of high concentration to an area of low. Eventually, the concentration gradient is going to be gone as all the sugar dissolves in the water, and the dye is, you can see, it's spread out now evenly throughout the solution. We actually call that an equilibrium, right? That would be no more concentration gradient. Okay, it's kind of, everything's just kind of balanced out, right? So eventually the concentration gradient is eliminated and the molecules are evenly distributed, right? That's what we would call an equilibrium. Right, so this is a good example of diffusion, where we see the movement of the molecules, again the sugar molecules, and we can see them because they put the dye, and we see the movement is from an area of high concentration. All this here has no sugar, right? And you can see that the cloud just starting to move away, get bigger, and eventually reach equilibrium. So what are some of the factors that influence diffusion? Meaning that what's going to cause diffusion to be either faster or slower. Number one is distance. As I mentioned earlier, the shorter the distance, the faster the diffusion. Right? If there's a big distance that something has to move, it's obviously going to take longer. Right? So the shorter the distance, the faster diffusion will occur. The ion or the molecule size, if something is smaller, it's going to move faster, right? It's kind of like weight. If something has a lighter weight, it's going to move faster than something that has a heavier weight. Same type of thing. Temperature, higher temperature, faster diffusion, right? So if we heat something up, that's going to cause the molecules to move faster. And if you just think about, boiling water or simmering water, as the water gets hotter and hotter, you can see the water moving, right? And when it simmers or when it starts to boil, That's because the heat is causing the molecules to move faster. So as a result, diffusion would be faster. Concentration gradient, again, that's the difference between the high and the low. The steeper the gradient, right, the higher the hill, the bigger the difference, the faster the diffusion, right? If there's a low hill, right, your bike wouldn't move as fast as if compared to if there was a high hill, right? You would go down that hill much faster. Same thing here. The steeper the gradient, right, the bigger the difference, the faster the diffusion. And then finally, electrical forces. Um, Remember the rule: opposites attract. All right. So if something is diffusing, say through the cell membrane, and there are opposite charges on the on either side of the cell membrane, that's going to happen faster. So opposites attract, while like charges repel. When things have the same charge, right, they're going to be repelled. So as a general rule, we'll learn this in more detail later in the semester. The intracellular fluid has a negative charge. So things that have a positive charge that want to come in will typically come in easier than something that has a negative charge, because negative charges are repelled. All right, so all of these are things that can have an impact on diffusion. The distance, the size of what's moving, temperature. Although in the body, temperature is pretty constant, so that's really not too much of a factor for us. The concentration gradient, and then finally electrical forces. So there are two types of diffusion that we want to study for the cell membrane. Highlight this first one here. It's called simple diffusion. Right? So let me read this and then we'll talk about it. This is the movement of, watch this, lipid soluble compounds, right? Things that are soluble in lipids, like phospholipids, for example. Movement of lipid soluble compounds across the plasma membrane. Things like alcohols, right? They dissolve in lipids. Fatty acids, steroids, like different types of hormones that are steroids. And then also, guys, dissolved gases such as oxygen and carbon dioxide. Here we go. So hopefully everybody recognizes this here, right? Phospholipid bilayer. We could see the different proteins that are embedded in the phospholipid bilayer. But here's the example. Things that are lipid soluble, such as lipid soluble molecules, even water to some extent, and soluble gases, oxygen and carbon dioxide, but steroids, that type of thing, they can easily move through the cell membrane. They just kind of diffuse from an area of high concentration. Let's say there was a lot of the steroid out over here. It can just easily pass through the phospholipid bilayer and come into the cell. Okay, and then again, same thing. If things were like carbon dioxide, let's say that carbon dioxide starts to build up on the inside of the cell. As it builds up inside the cell, it will easily just diffuse through the phospholipid bilayer and then leave the cell. Oxygen is going to be in a higher concentration out here because we're going to burn the oxygen on the inside, so oxygen will move in very, very easy. Okay, so anything that is lipid soluble and dissolved gases. Right, I didn't put it here, but water to a very small degree can move through the phospholipid bilayer, but you don't have to worry about that now. I just noticed here that they have it in the diagram. You can kind of ignore that. For me, I just prefer that you know that lipid soluble molecules and our gases, oxygen and carbon dioxide, they just move through the phospholipid bilayer. The second type of diffusion, if you want to highlight, is known as channel mediated diffusion. So this is the movement of ions and water soluble compounds, things that are not soluble in lipids. Now they have to move through a protein that we looked at early in previous lessons known as the channel protein. Right? These channels contain small little passageways. So let's take a look. If you notice over here, this is going to be a protein. It has a channel in it, and you can see water, right? Remember, this is the H2O. Here's showing like an ion, maybe it's chloride or sodium or something like that. These can, if they fit into the channel, they can move in based on its concentration gradient, or they can move out, right? So water, small water soluble molecules, right, not fat soluble, and ions which have a charge, right. Notice ions can't go through the cell membrane directly like the phospholipid bilayer. That they can't do. But if they have a charge, they can go through uh, this channel right here. Okay. Here they're also showing you too how the channels can actually block things from going through too if say something doesn't fit into the channel, right? So large molecules that cannot fit through the membrane channels will not diffuse through this method. We're going to see other methods that they will go through. Okay, so this is known as channel mediated diffusion. It has to be a protein that has a channel in it that will allow these ions to move along their concentration gradient. And again, both of these processes, guys, simple diffusion and channel mediated diffusion, are passive. They're just moving along their concentration gradient. Now, they call these channels that are just kind of open like this, like a tunnel, and it's just kind of open. We actually have a term for that. We call these channels leak channels. These are membrane channels which are always open, right? They don't close. They stay open. Kind of reminds me like a bridge or a tunnel that just doesn't have a gate on it. You can just drive through and then drive back out and drive through. Same type of thing. We're going to see proteins in a subsequent lesson where there are gates that the gate has to open in order for things to move through. Okay, so simple diffusion, channel mediated diffusion. Right, so that concludes this lesson on diffusion. In our next lesson, we'll take a look at the movement of water, which is kind of like a special type of diffusion that's known as osmosis.